You're watching CTOM TV. Knowledge is power.
I'm Michelle Morand, and this is Alexander Rowland, and we're co-founders of a worldwide independent research and advocacy group called Cancer Treatment Options and Management. We help cancer patients and their oncologists get access to leading-edge diagnostics and treatments that would otherwise not be a part of standard care for many years. We help them get access to them now. But what we're here for today is we're here to speak with Alex about what he and his team have been learning about COVID-19. As you can imagine, we're getting lots of questions from our clients and also questions from people who are not cancer patients about COVID-19 and all the information that we most need about, what it is, where it came from, what we can do to best protect ourselves, when vaccines will be coming along, and what current treatments are, and many other things. And so I prepared a list of questions for Alex to review and to provide answers on based on the questions that I'm hearing most from our clients. So today we're going to get Alex to answer all those questions as well as any other questions that you might have. So Alex, you have prepared some information based on your own interest and what you know we need to know, plus those common questions that we're receiving. Mm-hmm. So why don't I just turn it over to you.
Sounds good.
And you tell me what you've learned and what you think is most important for us to know right now.
Well, I think a bit of a primer on coronaviruses would be a great place to start.
Okay.
So coronaviruses were first discovered in the 1950s.
Right.
They're referred to as the order Nidovirales.
All right.
And they first found them in chickens that had infectious bronchitis. So bronchitis is not normally infectious, but this is a particular strain of infectious bronchitis that would wipe out these poultry farms. And if one chicken got it, almost all of the chickens got it. So literally, they would just have to kill all of the chickens and then spray down, the stalls and start from scratch.
Wow.
Years later, they started seeing it in other animals such as pigs and cows.
But we're going back to the 50s, you're saying?
Yeah, 1950s,
okay.
yeah. And what we know now is that coronaviruses have evolved with bats. Bats are the perfect vector, and bats carry, different types of coronaviruses. And bats are the perfect transportation vector because they are mammals, but they also fly.
So they get around a lot.
They get around a lot, but they're also mammals, so they
Right.
share a lot of the features that humans have. That we have.
Unlike
Primates,
birds.
unlike birds, exactly.
Right.
So they're a flying mammal, and SARS, which is SARS-1, COVID-19 is SARS-2. But SARS-1 originally developed, they believe, in civets and potentially in pangolins, which is a type of anteater. Another form, Middle Eastern respiratory syndrome, was MERS, and that went from bats to camels. And the death rate for that was 37%. So it was quite scary. So
shoot.
Yeah, so from bats to camels to humans.
And I know there was a MERS outbreak in Asia,
South Korea,
2015?
Was it South Korea?
Yeah. Among a few other places. So that's not so long ago.
Right. So coronaviruses have a long history with humans. We have a complex relationship. So if you'd like to put on your Facebook profile of your relationship with coronavirus, you could put complicated. It's complicated.
Long-term,
it's complicated. So there's at least 4 different strains of coronaviruses that cause about 30% of the common colds that we get every year. So every year we're all infected with these 4 different strains, or the vast majority of us are. However, they typically just affect the upper respiratory tract and the sinus in the throat. Whereas the SARS subtypes and the MERS, they affect the lower respiratory tract in the lungs, and that's what makes them so deadly. So, literally, why is COVID-19 so deadly? Well, it's because it has the infectiousness of a regular coronavirus that we normally see that causes colds, that normally only causes the problems in the upper respiratory tract, but it has the transmission. It's easily transmitted, but it has the deadly effect of infecting the lungs, such as SARS and MERS do. SARS-1 and MERS. So one of the reasons for this is it has something called a spike protein. All coronaviruses have spike proteins. But this particular version, COVID-19, the spike protein has a talent for binding and fusing with lung cells due to something called the ACE2 receptor. Okay. So the ACE2 receptor is a receptor in our cells. It's common in lung cells. It's in kidney cells and some other organs. But it provides a perfect opportunity for the COVID-19 to bind with and attach to lung cells, because it's a protein that's commonly expressed in lungs. So SARS-1, the original SARS, binded well to the ACE2 receptor. However, COVID-19 binds with 10 times more efficiency. So it's very, very effective, 10 times more effective at binding to this ACE2 in the lung tissue. The lungs. Yes. Now, unlike SARS and MERS, COVID-19 also uses a widely available enzyme called a furin. Now, this is a common enzyme in humans. Now, in a bit of a background, in order for the coronavirus to infect a cell, first off, what happens is the spike protein binds with the ACE2 receptor, and then it has to cleave it off at the stem of the cap, or the protein's cap from its stem. It needs to be cleaved off. And once that happens, it allows the membrane of the virus to bind with the membrane of the cell, and then it can spill its contents, the RNA, into the cell and infect it. So without that process, it's not able to do that. I see. So because furin is available in the lungs and is a common human protein, COVID-19 is adapted to use that, to cleave it. So it's able to infect us for a variety of different reasons. Number one, its higher affinity for the ACE2. And number two, its ability to use the furin to complete the next step. Now, once the virus is infected in our cells, then typically it uses our cellular machinery to copy itself until it reaches a certain number and it spreads out. Now, most RNA viruses, and this is an RNA virus, most RNA viruses like this typically have very poor replication abilities. And that is because they have a high mutation rate. So in other words, a lot of the progenitor viral particles will be, not viable because they're so mutated from the original one. Coronaviruses are unique in that sense, is they have something called a proofreading function. And so what happens is, unlike most RNA viruses, they're able to fix their DNA or their RNA. So this is something that humans have too. When we reproduce, whenever we get damage to our DNA, we have a series of different proteins and enzymes called DNA repair enzymes that will fix it and will put a checkpoint on it and prevent that cell from copying itself and producing daughter cells until that DNA is perfect and it's all fixed. Got it. So coronaviruses are able to do that too with their RNA. So they're very unique. In case of RNA viruses, they're very unique. They've probably developed and evolved this proofreading function because they have a very large genome for an RNA virus. So RNA viruses are usually not that large, but coronavirus has about 30,000 nucleotides, which is a fairly large RNA virus. And this overall allows it to remain stable in its host. This combination of having many different genes and proofreading function is it allows it to remain stable in the host when there is no selective pressure. It doesn't need to mutate. It doesn't need to change. But when it needs to, it can rapidly evolve and adapt to new species.
Hence,
Exactly.
That's what we're seeing here.
So one of the most important aspects of viral transmission is its ability to shed. And that is how its host releases the viral particles to infect other hosts. So viruses replicate in order to shed. and shed from its host. And then they shed their viral particles in mucus and snot and phlegm and even in breath, on molecules in your breath. So COVID-19 is a super shedder specifically in people that don't have symptoms. So in other words, when you get infected, if you don't have any symptoms, you are 55% more likely to be a super shedder. Or in fact, if you have mild symptoms, you are 55% more contagious as people with severe cases of it.
And why is that? How does that?
We don't know yet.
We don't know.
We're not sure. We just know that the symptomless people or the people with mild symptoms are super infectious. And importantly, in severe cases of people that require hospitalization on respirators and so on, in those patients, the virus can be still shed from those patients for up to 40 days after they've been infected. So, this is kind of an important thing that we're going to cover. So to give you some background on this, 100 million coronaviruses can fit on the head of a pin. It only takes about 1,000 to 10,000 particles to cause an infection. And they can live on, COVID-19 can live on air droplets for up to 3 hours.
Wow.
Some people are super infectors.
Right.
We haven't quite identified who and why.
Right.
We know that having low symptoms when you're infected means you're probably shedding a lot. So the bottom line is we need to test everyone. And that's going to be very, very important.
Yes, I'd say.
So I'm just going to get into some testing and what testing is all about for this. So if you could cue slide 1.
Oh, sure.
So the standard testing is what we call reverse transcriptase PCR, polymerase chain reaction. We were looking at 3 different proteins, but now we're looking at 2 different proteins. I don't believe CDC is suggesting we look at the spike protein anymore. So for COVID-19, it's 2 different proteins that we are concerned with. So the process is basically what we'll do is we'll collect the sample. So we'll collect a sample of it, and then we'll ship it off in a container, and it goes off to the lab, and then the lab will isolate the RNA. The RNA gets converted into cDNA, and then we do the polymerase chain reaction, which uses primers or sometimes color probes, such as TACMAN kits that use Vic and FAM antibodies to cause a light-based response when you have that. Okay. And then that's read, and or you can use a gel, traditional gel, and just look for those amplified proteins.
Okay.
So this is a laborious process. It requires a respiratory specialist lab, and this is people who are well protected to take the samples. So you can't get tested in your regular doctor's office as you do with a standard influenza or a standard process. You can't go to the hospital and get tested. You have to have a specialized crew to test you. And then, of course, the transport of live viruses is going to be prohibited across borders. So you can't really just use testing facilities anywhere in the world. You have to use testing facilities in your own country. And then, of course, transporting these samples, they have to be on ice. It's a very costly, laborious process. And, it's just not the best way. So currently, we're looking at a spread of 8 different substrains of coronavirus. So here's a map right here of the different strains and how they've spread. So Michelle, if you'd like to play it, you can see where it all started. This is a timeline-based spread of the coronaviruses. So as you can see, it started in Wuhan. It's getting larger. Starting to spread. And typically it's being spread locally, and then it's being spread through air traffic.
Yeah.
And so initially it was one strain that was spreading, and then there was some mutation that happened. And as it spread, these different mutated strains started to spread.
Mm. Wow. So you see the Italy area where there was so much concern, and still has more yellow.
Yeah, yellow and green.
In North
Whereas we
America.
have more of a red colored strain.
Mm-hmm.
So yeah, 8 different strains right now.
Mm. Wow. Mm.
And this is where we are today.
Well, not exactly.
Not yet.
Yeah.
Is it frozen?
No, that's it.
No, it's not it. It should keep going.
Oh, no.
It's frozen.
Mm.
Oh, that's too bad.
Well, actually, I think we saw enough there. I can certainly press play as you move on and talk about other things, but
we're going to go to a different slide now.
Oh, okay.
Yes.
Well, I will pause and we can come back, folks, and I'll let that play while Alex is sharing with us. You want to see this
the
one?
sequence divergence. So this is where this is a timeline of the different sequences.
Just going to set it up again.
This is a timeline of the different sequences, and from left to right, as you can see, December 3rd, everything's sort of purple. And then the strains started progressing as you go to the right. Mm-hmm. And what this shows that the red strain that we have over here in North America didn't really evolve until January 29th, just before January 29th. Mm-hmm. So it's a fairly unique strain that we have over here. Mm-hmm. So definitely 8 different strains, maybe more. So what this boils down to is, you need to know the status of the infected and the uninfected, and you need to know which strain you have. I think this is a very important thing. So we're going to talk about testing. Obviously, home testing is one of the best options. Okay. Antibody testing is the way of testing for antibodies that are unique to. Can you go to the 3rd slide, please?
Oh, sure.
Antibody testing is there we go. Is the best way to test right now for knowing if you've had exposure to the disease.
And can you tell with antibody testing whether you've had which of those 8 strains you've had?
No, it just tells you that you've had exposure to COVID-19.
Okay.
And basically, what they do is their antibodies that your body produces called IgG and IgM in response to it. And so it's much like a home pregnancy test or blood glucose monitoring test. Mm-hmm. It tells you if you have antibodies that are developed to previous infection. And so it typically takes about 14 to 21 days for you to develop these antibodies against COVID-19.
Okay.
But however, people can remain infected for a long period of time after they develop antibodies. And this is unique to COVID-19. So typically when it takes you about, 14 to 21 days to develop antibodies. But when you develop COVID-19, just because you have antibodies, the assumption is that, okay, now you've got antibodies, you're over the infection, your body's gotten rid of it, your antibodies are proof that your body's got rid of the infection. However, this is not the case with COVID-19. And in fact, people are testing positive for the virus, 6 weeks after they've produced antibodies.
Whoa.
So,
Wow. So normally it'd be like 14 days. You start
almost 6 weeks. Yes. So the key here is you get tested. Then if you have antibodies, you wait 6 weeks and get tested with a PCR or sequence-based test to ensure that you don't have the virus anymore. So all of this stay home for 14 days and then you're, virus free and you're not infectious is just misinformation. And this is the reason we have this pandemic right now. So unfortunately, the FDA has stopped allowing self-testing. They won't allow home-based point of contact testing for patients at this time. And that's unfortunate because there's about 5 or 6 companies that are producing these antibody tests. They're very cheap. People can use them in their home. And there's a variety of reasons. The FDA wants to make sure that people aren't going to be assuming they're negative when they could be positive, or vice versa. They want to oversee this. I think it's kind of a bad idea, though. I think all testing needs to be done, and I think this is kind of very relevant. So probably the most important type of testing is called sequencing. And so if you look down at your page here, and you see this is a coronavirus, this is a composite coronavirus sequences. Now we're missing a bunch of mutations. I think, can you resize that, Michelle?
Yeah, I'm just going to start again here and see.
Go all the way down. All the way down. All the way down, right there.
Yeah.
Yeah, so for some reason, we're missing a bunch of mutations here.
Do I need to set it there?
No, let's not play with it.
Oh, too late. I've already played with it. Yeah, I don't know what's happening here.
Okay, just go down to it, please.
Okay. Oh, hang on. 1 second, everybody. Oh, what is that? Why is that happening? There we go. Okay.
Okay, so right here, there's a lot more mutations here, but if you put your mouse, Michelle, could you go to the right and put your mouse over one of those spikes?
Sure.
These are the different positions of the mutations. So this is codon 84 of open reading frame 8. And as you can see, because it's an RNA virus, please go back to it.
Oh, sure. Sorry about that.
As you can see, this is a mutation that occurs in some strains, and it's 2 nucleotides, 28143 to 28145, because RNA viruses require 3 different nucleotides to code for an amino acid. So there's some functional redundancy in amino acid coding. It's not the same as DNA, so you need 2 different variants there. So sequencing allows you to determine the different strains. So how about treatment? What can we do for treatment? So there are 2 drugs right now.
This is just to back up the bus. The reason we want to know what strain we have is because potentially there's a difference between how they're affecting people.
Exactly.
And and this treatment, of course, might vary based on the strain.
No, probably not.
No, probably not.
Vaccines will vary based on the strain.
Okay.
So there are 2 RNA targeting drugs. One's called remdesivir. It targets the proofreading function of coronaviruses. And so it's very effective. We've seen patients who are on ventilators cured within a few days and released from hospital, where they probably would have died without this drug.
Wow.
The Chinese have done clinical trials of it, and they found it's incredibly effective.
It's amazing how quickly it works. Not just that it works, but
and that's because it interferes with the proofreading. So all of a sudden, the coronavirus is producing non-viable offspring. There's another drug called favipiravir. It creates mutations in the coronavirus, and it also affects the polymerase that transcribes the proteins. It's also quite effective. So these drugs really are excellent for treating. They're new drugs. Well, they've been around for a little while, but there's not a lot of them because there hasn't been a lot of interest in coronavirus. So, I mean, if we could have access to these drugs, we could definitely help a lot of people. Also, because coronaviruses require the furin enzyme to cleave off the spike protein so the virus can enter the cell, or the RNA protein can enter the cell. Furin inhibitors are probably going to be quite effective with this.
And those are readily available?
Not yet.
Oh.
Yes. Well, not specifically for coronaviruses.
Okay.
So there's also vaccines in development. We're going to need a multivalent vaccine. It's probably going to be much like seasonal flu, and they're probably going to be changing it every year. Now, this virus is only mutating at about 25 mutations per year. But obviously, we have different strains going on, and we don't know which mutations are going to cause the ability for it to reinfect. We don't know what's going to happen every year. We know this virus has been with us for a while, and it's only recently become highly pathogenic. But I think we need to really do a lot of sequencing. And I think the sequencing and, tracking, seeing what's happening in the different strains is really going to be an important thing.
Mm-hmm.
So what do I see in the future? Well, I mean, we've got 4 different variants of coronaviruses that cause flus and don't kill us, or not flus, but common colds, and they don't kill us. They're not problematic. They've evolved with us. I suspect COVID-19 is one of those. I suspect it will be one of those. And it's just trying to adapt to us using whatever abilities it can. Typically with RNA viruses and other viruses, the deadly strains burn out after time. Otherwise they kill their host and they can't multiply, and that's it for them. They need us. We're important for them. Also, viruses tend to adapt to the host and become less dangerous over time. And in that sense, they also are able to better evade the immune system. And that's common with, things like HPV and, different viruses that have lived among us. Epstein-Barr, for example, where most people have infection of some sort, but it doesn't typically kill you. So that's where I see this virus going. I think it will become less pathogenic over time. We don't know that for sure, but we're not going to survive if it doesn't, because it's highly transmissible.
Right. Okay. So right now, if my notes are accurate, we have this kind of coronavirus has been amongst humans for quite some time in different forms.
Not this strain, COVID. We're talking COVID-19.
Right.
There's many coronaviruses.
Yes, thank you. That's what I'm just recapping. As you talked earlier about how coronavirus isn't new to us, but COVID-19 is.
It's a pathogenic strain that's new.
Yes.
Although it's the COVID-19 strain itself has probably been with us for a while. It's probably been around since the 70s or the 80s.
In humans.
In humans, it probably just hasn't been as pathogenic and as transmissible as it is now.
Right.
So it hasn't just recently jumped to humans.
Right.
The data shows that it's been around for a while, and that it's recently become more pathogenic and more transmissible.
Okay. And partly because it's got this special function of being able to proofread itself and therefore catch any mutations.
Well, all coronaviruses have proofreading.
Oh, do they?
Yes. It's part of how they maintain their large genome. They have a huge genome with 30,000 base pairs.
I thought that was unique to COVID-19,
No,
but it's coronavirus in general. Yes. Okay, so I see that's problematic. But you're saying there are some treatments available right now. They're just not as we'll say multiple as we need, or as easy to access. The drugs just are not
available. The people are getting them, but they're kind of lucky.
Right. So they are. Just so he's a scientist, he speaks a different language from me. These things are available. It's just that there's not enough of them just yet.
Exactly.
So obviously, pharmaceutical companies and medical services companies are working hard to create more of these antivirals and these furin inhibitors that you mentioned. You also talked that almost certainly there'll be some sort of seasonal vaccine that we end up with, hopefully sooner rather than later, akin to our seasonal flu vaccine. So that is something definitely to look forward to, that it's possible, very, very, very probable to develop a vaccine, and for that to be something that becomes one of our annual shots that we get to prevent this kind of pandemic in the future. And you were talking about tests. There's 2 different kinds. At least right now, there's
3 different types.
3 different types of tests. So there's the sequencing test, and that would tell you, if I understood you correctly, that would tell you if you had active coronavirus right now.
And which strain.
And then there's the antibody test, which would tell you if you've had it. But with this COVID-19, you're telling us that what's special about this is that the period of time from the time you're infected to the time you develop antibodies and then you're no longer infectious is much longer than a standard.
It's probably going to be 2 to 3 months.
Right. Whereas normally, as soon as you start to develop antibodies, you start the clock for 14 days, and then you're no longer contagious.
Infectious, exactly.
Right. Whereas with this, one of the problems is, aside from how easily those asymptomatic folks, which is a large percentage, how much they shed the disease or the virus, but then also it's at least 40 days from the time you start to develop antibodies to the time that we're still seeing people having this live virus.
Exactly. And you could carry the virus on your skin and not infect yourself for, a week or two.
Wow.
That's possible too.
That's interesting. So really, we have to be more careful than we realized, and that period, that incubation period, or that period that saying that differently, the period that we need to sequester ourselves if we think that we have any likelihood of having been infected and been one of those very low symptom or asymptomatic people, is almost certainly 40 to 60 days as opposed to just 14.
Yeah, at least.
Whew. So here we come back to these tests. Being able to have one of those antibody tests so that you can at least start the clock for those 40 to 60 days.
It's very important.
Would be very
integral part. And I think, one of the biggest problems is that the people who are running this, overseeing this pandemic, these medical doctors, just don't have the scientific background and knowledge of coronaviruses to be able to understand what needs to be done.
Possibly, yes. I suspect, but given the nature of this, I think that it would be wise to assume that there's lots of scientists who are influencing the doctors on the front line and working together for sure.
Yeah, there's been almost 1,000 different peer review studies lined up right now, being peer reviewed right now. Just in the last 2 months on coronavirus, on COVID-19.
All right. So the scientific community has prepared a lot of information, and as we find in our work with cancer, there's often such a delay from the time something is proven, peer reviewed medical science, and even FDA approved, to the time that's available to the folks on the front line. With cancer tests and treatment, that can be anywhere from 2 to 10 years. With something like this, obviously, it's not going to be that long. It's just that we do now have this information bottleneck with these 1,000 or so papers that still need to be reviewed before they can be properly published, and then the doctors on the front line can read them and find out about what's possible. So obviously making sure that you are paying attention to reputable news sources, really Johns Hopkins in North America has an excellent page about COVID. I would highly recommend you focus there. World Health Organization, the UN are both providing very good information about what's happening. And the Canadian government, where we are, is doing a fine job of keeping people updated with some excellent fact-based science. So there's some really good resources, and I encourage you to check those out. And that image that we shared with you earlier, that from Next Strain, that's just helping you to understand how many different strains of this virus are, and that map that shows where it began and how it's transmitted across the world, but also how it's changing as it's going. That would be a good site to bookmark and come back to from time to time, just to see, how the virus is mutating, or how new strains are coming along.
Exactly.
And again, what's most important is to be keeping an eye on what those strains are telling us, how different they are and how differently they're affecting people. And we'll certainly update you on that as we have more news. And keep an eye out for antibody testing and for that sequencing testing, because you'll want to have both. You'll want to make sure that you find out if you've had those antibodies, and then you'll want to have that COVID-19 sequencing test to find out if the live virus is still present in your body. And then you'll be able to breathe that sigh of relief, and you'll know you're not contagious, you're not going to infect anybody else, and you'll be able to go back to business as usual. If you'd like to find out anything about COVID-19 testing, what are the best ones, how to get a hold of them, and the antibody testing, you can contact us through our Facebook page. You can send us a contact form at CTOAM, that's Cancer Treatment Options and Management.com, and we'll make sure that you get the answers that you need, and we'll keep you posted. Thanks for joining us today.
Thank you for watching CTOM TV. Subscribe below and stay informed.